Students, today we study process of abscission, how abscission work. The topic belongs to plant anatomy. First of all, we study what is abscission. Abscission is the separation of leaves, flowers and fruits from plants after the formation of an abscission zone at the base of their petioles, peduncles and pedicels. Abscission, a physiologically determined program of cell separation. Abscission is the separation of leaves, flowers and fruits from plants after the formation of an abscission zone at the base of their petioles, peduncles and pedicels. Abscission, a physiologically determined program of cell separation, provides a mechanism whereby every discrete multicellular plant organ such as leaves, flowers or fruits becomes detached from the plant body in a controlled manner. Abscission can be initiated in response to environmental events like such as a disease or pathogens, or it can be programmed shedding of organs that no longer provide essential function for the plant. For example, the flower after aiding in pollination, the process requires the formation of an anatomically distinct structure, the abscission zone, which constitutes the region where organs are detached from the plant body. Very little is known about which development signal and how cell-cell interaction in formal primordial age Z cells to differentiate. The diagram shows the abscission zone between pedicel and stem. The darker cells, thickened and darker cells, are the abscission zone cells. Structural mechanism of abscission zone. In deciduous trees, an abscission zone, also called a suppression zone, is formed at the base of the petiole. It is composed of a top layer which has cells with weak walls and a bottom layer which expands in the abdomen, breaking the weak walls of the cells in the top layer. This allows the leaf to be shed. Lack of chlorophyll is a trigger mechanism for the abscission. The reduction of chlorophyll production in leaves due to decreased sunlight in the fall explains why some leaves turn yellow. However, the yellow color can attract aphids, so some trees turn the leaves red instead by injecting a bright pigment. The loss of chlorophyll may also contribute to the abscission process. The diagram shows the abscission of the hyphaenthum during development of a nectarine fruit. Abscission zone. The abscission zone is a layer of weak, thin-walled cells that form across the base of the plant part, where the break eventually occurs. A corky layer containing suberin forms beneath the abscission zone to protect the plant. The process occurs at a precise sites and involves coordinated cell wall breakdown. Associated with cell separation is an increase in the activity of several hydrolytic enzymes, including like 1,4-glucosinase, EC and polygalactonuclease. The diagram shows the abscission zone between leaf side and tree side. The process of leaf loss is called abscission. and is controlled by the hormonal activity. The abscission zone is located at the base of the petiole in a region of undifferentiated small parenchyma-like cells. Their walls contain no lignin, and the vascular cells in the abscission zone are also reduced in size. The process of abscission is initiated and proceeds as follows. The first one is the parenchyma cells start dividing rapidly. They secrete a layer of suberin in the walls nearest the stem. The middle lamella, cell walls, and the cells of the abscission zone dissolve due to the enzymatic degradation activity. Leaf abscission, the last step. The process of leaf abscission. The abscission of leaves is a highly coordinated phenomenon involving multiple changes in cell structure, metabolism, and gene expression. The diagram shows the abscission zone during the process of leaf abscission. Induction of abscission. There are a variety of environmental factors accelerate abscission. Mineral deficiency, drought, low light as being responsible for the shedding of the flower and fruit. Pollination, accelerated petal abscission and failure of embryonic development seemed associated with the shedding of young fruit. Both environmental and natural factors influence the rate of natural abscission. Various phytohormones role in abscission. Cause the abscission of leaves and fruits. Abscisic acid was found to be an inducing factor. Accordingly, later it turned out the formation of the abscission layer for leaves and fruit is induced primarily by ethylene. Auxin is another plant hormone and ethylene have been implicated as prominent regulators of abscission signaling. The two compounds work in a synergistic fashion. As the IIA level decreases, the flux of IA2 with the abscission zone is reduced. Exhaustion of IA makes the abscission zone sensitive to ethylene. When the plant is then exposed to ethylene, gene expression of cell wall degrading enzymes such as cellulose and polygalactonurease are activated. However, this is not to say that ethylene directly activates WD gene expression. Why researchers originally believed ABA to be the hormone that stimulated abscission process. Auxin levels have also been implicated in autumnal leaf color change. Auxins have often been reported either to delay or induce fruit abscission. The diagram shows the leaf abscission, progressively leaf senescence process or formation of abscission zone in leaf petiole. Role of abscisic acid in abscission. ABA also promote abscission of leaves and fruits in contrast to auxin, which inhibit abscission. It is in the fact that the action that gave rise to name abscisic acid. The drooping of leaves in the atmosphere is a vital response to the onset of winter when groundwater is frozen and thus cannot support transpiration and snow load would threaten to break any branches still in leaf. Role of gibberellic acid and cytokinin in the process of abscission. Both gibberellic acid and cytokinin will influence abscission, although they are thought to be the less important. Then the other are plant hormones. Cytokinins can delay abscission, probably by indirectly. Delaying senescence, gibberellic acid will accelerate abscission. Control of the abscission process. Abscission is controlled by the hormones auxin and ethylene. and that it is also closely correlated with environmental factors such as photoperiod process. Ozone wounding and attack by pathogens, water stress and senescence are the other factor which can control the process of abscission. Control mechanism for the process of abscission. Oxygen is a primary growth regulator produced in the leaf and slowly transported towards the stem base through the living cells. As long as oxygen is effectively being transported across the abscission zone cells remain unreactive. As oxygen production begins to wane and fall and oxygen transport rates begin to decline due to less oxygen availability, damage to living cells transporting oxygen and oscillating infection of living tissue bypass, cell wall changes are initiated. Cell wall changes increasingly inhibit oxygen transport and accelerate ethylene production. Small amount of ethylene hasten abscission zone development. ABA responsible for dormancy. Onset on the leaf stimulates ethylene production and inhibit onset in the leaf stimulate ethylene production and inhibit oxygen transport. That's all about some facts about abscission and leaf abscission.